Of all the major multicellular groups of organisms, what used to be known as the kingdoms, fungi and plants and animals, the animals are the youngest. They're a whippersnapper, only about 1.2 billion years old. What's shown here is all of the phyla that are within the animal, what used to be known as the animal kingdom is now generally referred to as the metazoa. The phylum is a major taxonomic grouping. The plural of that is phyla, and you'll be glad to know that we are not looking at all of these phyla in the next 4 lectures. These are the ones that we are going to be looking at. Each one of these is a phylum, and together they tell a very interesting story about evolution. They tell an interesting story about the evolution of animals in particular, but there are also some very interesting lessons to be learned from a study of the animals about principles that apply to life in general. And we'll be seeing how some of those things play out over the next few lectures, of course. So what are animals? Animals are all multicellular, and of course they are eukaryotes. All of them are heterotrophs. That is, they get high energy carbon compounds from other organisms. Basically, they eat other organisms or in some way consume stuff made by other organisms in order to get their carbon and energy. There are a lot of animals that do not move, but animals have a capacity to move and behave that is much, much more developed than what you see in any of the other organisms on Earth. They can be very simple, as we'll see later in this lecture, but they can also be extremely complex. If you measure complexity based on the number of cell types or the number of tissue types that are present in an organism, the most complex organisms on Earth are animals. There are a couple of features that are, or actually tissue types, that are unique to the animals. And these are, of course, intimately related to their ability to behave in really complicated, sophisticated ways. Muscle tissue is a type of tissue that's only seen in animals. And the nervous system, nerve tissue, is only seen in animals. Now, there are other organisms that move, and some of them can move pretty rapidly using other mechanisms. And there are even organisms that send electrical signals within their bodies. However, none of those organisms match the complexity and the speed with which animals can take in information, integrate it, and turn that integration into behavior. There are a tremendous diversity of forms within the animals. There are organisms that within this group that you recognize very clearly as being similar to yourself. There are others that look like aliens, and there's still others that for centuries were actually classified as other things like plants. They look very, very different one from another. Sadly, we can't cover all of the diversity of the animal kingdom in these few lectures, but I do want to give you a taste of what is out there. Before we start actually talking about the different animal phyla that you're expected to know in this course, I'd like to talk about some basic principles about the organization of animals, because it may be useful to have certain terms and concepts down in your head before we start looking at different animal forms. One of the things that's very fundamental about any organism, but this is particularly interesting in the animals, is its symmetry. There are some animals that have no symmetry. So for example, with sponges, no matter which way you look at them, you don't get any mirror images where one half is exactly the same as the other half, just the mirror image of it. Okay, there are many animals that have what is called radial symmetry. That is, so for example, if you look from the top down on this hydra, there are a number of different planes along which you could cut that in half, and you would essentially get two mirror image halves. The same thing is true for the sea anemone over here. It doesn't really matter which plane you look at, you've basically got two mirror image halves. There are some other variations on radial symmetry. So for example, a sea star is not exactly radially symmetrical. It's what's called pentaradially symmetrical. That's a relatively minor variation on this radial symmetry. The one that's really different than these two that I've shown you already is bilateral symmetry. Okay, in bilateral symmetry, there's only one plane, and that's what's shown here in blue, along which you can divide an organism and have the two halves basically be mirror images. You can see bilateral symmetry quite clearly being different than radial symmetry. If you were to try to split this alligator any way other than this plane right here, you would not get two halves that looked that were mirror images of one another. Also, it would eat you. Along with different kinds of symmetry, there's another pattern that sometimes shows up in the animal kingdom, but again, is not found in all animals. Cephalization basically means it's the act of having a head. If something is cephalized, it has a head end and a tail end. Technically, what cephalization is, it's when the sensory organs and usually nervous tissue are all concentrated up toward one end of the body. And clearly, that is seen in some animals and not others. Generally, cephalization goes along with bilateral symmetry. And there is a reason for this that we will get into probably in the next lecture. On the inside of animals, there are a few structural themes that you should know about. An animal is not just a solid mass. For one thing, they usually have a digestive tube that runs down the middle of them from mouth to anus. But wrapped around that tube is usually a cavity that is called a coelom. This is not the digestive system. This is something that is distinct from the digestive system. What's shown over here, this is an earthworm or an annelid. This is what a coelomate animal looks like, an animal with a true coelom. During development, the middle tissue layer, the mesoderm, splits open so that this hollow space opens up within it. What's shown in yellow here is the endoderm, and the red is the ectoderm. The endoderm goes on to form the gastrointestinal system in the organisms that are most familiar to us. The ectoderm forms the outer covering of the animal. So the equivalent here is that if this were your stomach or your intestinal system, and this were your skin, this area in here would be your peritoneal cavity. So if someone took a cross-section of you across the middle, you see the skin on the outside, the gut tissue in the middle, but then there would be this opening that is not the gut, and that's called the coelom. Why does this matter? This actually has some very important implications for the way that organisms work, but it's also a really fundamental division in the way that animal bodies are put together. There are some organisms that have no coelom, like this flatworm. This is what's called an acoelomate animal. There's some that have a pseudocoelom, which is actually a gap between the mesoderm and the endoderm. The roundworms are pseudocoelomates. And then there are true coelomate animals like the earthworm. While we're speaking about the digestive system, digestive systems actually come in a couple of different flavors, no pun intended. There are incomplete guts and there are complete guts. An incomplete gut is a blind sac. So for example, a sea anemone has one opening to the gastrovascular cavity. That's its digestive system. This opening is therefore both the mouth through which food comes and the anus through which food is expelled. Now, that may sound like a horrible concept, but hopefully it'll just help you remember it. What we're more accustomed to is what's called a complete gut, where there's a separate mouth and anus, and material generally moves only in one direction. Within the animals that have complete guts, there's a further division. There's a group of organisms called the protostomes, and there's a group of organisms called the deuterostomes. Proto means first, stome means mouth. And so the protostomes are the animals where the mouth forms first in development, and then the anus forms later. The mouth actually forms from the blastopore, which if you remember anything from developmental biology, this isn't going to be on the exam in this class, but you might remember it. The blastopore is the first pore that kind of lets into the hollow blastocele. In protostomes, that becomes the mouth, but in deuterostomes, that actually becomes the anus, and that opening forms first. A second opening occurs later in development, that becomes the mouth. So again, this is not the sort of thing you can see on the outside of an animal, but it's it's a difference in evolution that happened very, very early. And it's a signal or a sign that this whole group of organisms over here shares common ancestry, whereas this whole group of organisms over here shares a somewhat different common ancestry. And we'll be hearing a little bit more about the protostomes and deuterostomes later on. Another theme that actually shows up in some unrelated groups within the animals is segmentation. The technical term for this is metamerism. But this happens when you have repeated body elements. You see this in the segmented worms, perhaps obviously. You see this in certain arthropods. In fact, it's present in all arthropods, but it's much more visible in some than others. And you see it in vertebrates, like this turtle.